Resistance and the immune system. Innate immunity. Immunology is the scientific study of how the immune system functions in the body to prevent or destroy foreign material, including pathogens. In the circulatory system, blood consists of three major components: the fluid, the clotting agents or platelets, and the formed elements or cells. The fluid portion, called serum, is an aqueous solution of minerals, salts, proteins, and other organic substances. When clotting agents such as fibrinogen and prothrombin are present, the fluid is referred to as plasma. Blood cells form an important defense for innate and acquired immunity. Blood platelets are small, disc-shaped cells that originate in bone marrow. Platelets have no cell nucleus and function chiefly in blood clotting mechanisms. There are 6 major types of white blood cells: the granulocytes, which include neutrophils, eosinophils, and basophils; the monocytes, which includes macrophages and dendritic cells; and the lymphocytes, which includes B cells and T cells. Neutrophils have multi-lobed cell nuclei and therefore are referred to as polymorphonuclear cells. Their cytoplasm contains many granules that contain digestive enzymes and other antimicrobial agents capable of killing pathogens. Neutrophils function chiefly as phagocytes. Phagocytes are cells that carry out phagocytosis. Neutrophils can move out of circulation to engulf foreign particles or pathogens causing infection. Eosinophils exhibit red or pink cytoplasmic granules when a dye such as eosin is applied. Substances in the granules contain cytotoxic proteins that defend against multicellular parasites, including flukes and tapeworms. Eosinophils are involved in the development of allergies and asthma. Basophils are leukocytes whose cytoplasmic granules stain blue with methylene blue. Basophils, along with their counterparts in tissues, which are the mast cells, also function in allergic reactions and defenses against parasites. Monocytes are phagocytic cells that have a single bean-shaped cell nucleus and lack visible cytoplasmic granules. In the tissues, monocytes mature into macrophages. In contrast to the 2-week lifespan of monocytes, macrophages may live for several months. The word macrophage means big eater. Macrophages are more effective as phagocytes than neutrophils and are one of the key cells in both innate and acquired immunity. The lymphocytes migrate from the bone marrow to the lymph nodes after maturation. They have a single large nucleus and no granules. One cell type, the natural killer cells, plays a key role in innate immunity by destroying virus-infected cells and tumor cells. The 2 other types are B lymphocytes and T lymphocytes. B and T lymphocytes are key cells of acquired immunity. They increase in number dramatically during the course of an infectious disease, but they have very different functions, as will be described in a separate discussion on adaptive immunity. Dendritic cells are found in the skin, where they are called Langerhans cells, and in tissues where pathogens might enter. As active phagocytic cells, they are crucial to innate immunity and the activation of acquired immunity. In the human body, the clear fluid surrounding the tissue cells and filling the intracellular spaces is called tissue fluid, or lymph, meaning water. Lymph bathes the body cells, supplying oxygen and nutrients while collecting wastes. The primary lymphoid tissues is where the lymphocytes mature, differentiate, and divide. The primary lymphoid tissues consist of the thymus and the bone marrow. The secondary lymphoid tissues are sites where mature immune cells interact with pathogens and carry out the acquired immune response. The spleen contains immune cells to monitor and fight infectious microbes entering the body. The lymph nodes are secondary lymphatic tissues that are prevalent in the neck, armpits, and groin. Lymph nodes carry macrophages, dendritic cells, and lymphocytes. Macrophages and dendritic cells engulf pathogens in the lymph. Lymphocytes respond specifically to foreign substances in the circulation. The lymph nodes become enlarged, often called swollen glands, during infection. Nonspecific resistance is referred to as innate immunity because genetically encoded molecules present in the body from birth are capable of recognizing microbial features common to many pathogens and foreign substances. The human lymphatic system consists of lymphocytes, lymphatic organs, lymph vessels, and lymph nodes located along these vessels. Upon recognition of microbes by the innate immune defenses, these defenses try to eliminate or hold an infection in check while sending chemical signals called cytokines to the tissues involved, with initiating the acquired immune response. This is called acquired immunity because the response produces lymphocytes and antibodies that are specific to only the pathogen or foreign substance that is causing the infection. The innate immune response includes physical barriers, chemical barriers, and cellular barriers. The physical barriers include the skin and the mucous membranes. In the skin, the keratin forms the outer layer called the epidermis. The epidermis forms a tough and impenetrable barrier to infection. In addition, the thin outer epidermis is constantly being sloughed off, and with it goes any attached microorganisms. Cuts or abrasions are a fact of everyday life. These breaches to the skin may allow microbes to enter the blood. This can occur through bites from infected arthropods that may inject pathogens responsible for such diseases as West Nile fever, malaria, or plague. Other means of penetrating this mechanical barrier include puncture wounds, tooth extractions, burns, shaving nicks, war wounds, and injections. The mucous membranes, or mucosa, are the moist epithelial lining of the digestive tract, urogenital tract, and respiratory tracts. Because these exposed sites represent potential portals of entry, the mucous membranes provide resistance to microbial invasion. For example, the cells of the mucous membranes that line the respiratory passages secrete mucus. Mucus is a sticky secretion of glycoproteins that traps heavy particles and microorganisms. The epithelial cilia then move these microbes along with the mucus up to the throat, where this material is then either swallowed or coughed out, eliminating them from the body. Several host chemicals also provide antimicrobial defenses to infection by inhibiting microbial growth or viral replication. Resistance in the vaginal tract is enhanced by low pH. In the urinary tract, the slight acidic pH of urine discourages pathogenic growth, and the flow of urine flushes the microbes away. Stomach acid, which has a pH of approximately 2.0, destroys most pathogens. Antimicrobial peptides called defensins are found in various secretions throughout the body. The oil called sebum on the skin's surface produces defensins which kills pathogens through cell lysis. Lysozyme is found in human tears, mucus, and saliva. The lysozyme enzyme disrupts the cell walls of bacterial cells, causing death. Complement components and interferon create physiological barriers to prevent infection. The blood monocytes, tissue macrophages, and neutrophils are key cellular defenses of innate immunity. These cells can carry out phagocytosis and trigger an inflammatory response to destroy invading pathogens. The normal microbiota of the body surfaces form a cellular barrier by outcompeting pathogens for nutrients and attachment sites. Here is a concept map summarizing the ways innate immunity is poised to inhibit, eliminate, destroy, and compete against invading pathogens. Phagocytosis is the capturing and digestion of foreign particles. If a bacterial or viral pathogen breaches the exterior defenses, the intruder usually is recognized, ingested, and killed by neutrophils or macrophages that are residing in the tissue. Upon binding the pathogen, phagocytes release cytokines that will trigger an inflammatory response, as well as activate an acquired immune response. Some cytokines, called chemokines, stimulate the migration of neutrophils to the infection site. Let's look at the mechanism of phagocytosis. In phagocytosis, the macrophage attaches to a bacterial cell as the plasma membrane surrounds the cell. The bacterial cell is then internalized into a phagosome. The lysosome then fuses with the phagosome to form a phagolysosome. Within the phagolysosome, the bacterial cell is killed and digested through the activity of lysosomal enzymes and other toxic products. At the end, the bacterial debris is eliminated during egestation. Neutrophils are usually the first type of cell arriving at the site of the infection. Because neutrophils are short-lived cells, the longer-lived macrophages arriving at the site are more likely to kill the majority of pathogens. While living, neutrophils contact pathogens. Some of these cells may transform themselves into an extracellular fiber network called neutrophil extracellular traps, or nets. In neutrophil extracellular traps, or nets, the neutrophils become deformed, and the nuclear envelope and granule membranes disintegrate. When the plasma membranes of the dying neutrophil ruptures, the net components containing nuclear DNA and granule antimicrobial proteins mix together. The invading pathogens get caught in these net fibers, and the antimicrobial agents degrade and kill the pathogens without the need for phagocytosis. However, during the fierce phagocytic activity occurring at the site of infection, lysozyme enzymes and other products may be released inadvertently into the surrounding tissue, causing damage. Pus, for example, is composed of dead and dying neutrophils and damaged tissue arising from the site of infection. Inflammation is a nonspecific defensive response by the body to trauma. It develops after a mechanical injury, such as an injury or blow to the skin, or from exposure to a chemical agent, such as an acid or bee venom. An inflammatory response also may be due to an infectious pathogen. In the process of inflammation, a series of nonspecific steps generates the inflammatory response. Here is an example. A thorn pierces the skin, causing a mechanical injury and bringing bacteria into the tissue. Tissue macrophages initiate phagocytosis and secrete cytokines. Cytokines cause the capillary walls to dilate, becoming leaky. Then, plasma flows to the site of injury, making it red, warm, and swollen. Phagocytes, which include the neutrophils and monocytes, attach to blood vessel walls. After this, the cells migrate into the surrounding tissues, where they arrive and begin phagocytosis. Monocytes differentiate into macrophages. Mast cells also arrive at the site and contribute to the immune process. As bacteria are eliminated, a fibrin wall accumulates around the mixture of plasma, leukocytes, bacteria, and tissue cells, forming a blood clot. Pus may also be produced in a cavity called an abscess. Finally, lancing of such an abscess releases the pus. A fever is an abnormally high body temperature that remains elevated above the normal 37 degrees Celsius. Fever supports the immune system response in trying to gain an advantage over the pathogens by making the body a less favorable host. Fever-producing substances called pyrogens are produced by activated macrophages and other leukocytes, and exogenous microbial fragments from bacterial cells, viruses, and other microorganisms. Pyrogens move through the blood and affect the anterior hypothalamus such that the body temperature becomes elevated. As this takes place, cell metabolism increases and blood vessels constrict, thus denying blood to the skin and keeping its heat within the body. Patients thus experience cold skin and chills along with the fever. A low to moderate fever in adults may be beneficial to immune defenses. This is because the elevated temperature inhibits the rapid growth of many microorganisms. A low-grade fever also encourages rapid tissue repair and heightens phagocytosis. However, if the temperature rises above 105 degrees Fahrenheit, convulsions and death may result from host metabolic inhibition. Also, infants with a fever above 100 degrees Fahrenheit or older children with a fever above 102 Fahrenheit may need medical attention. Natural killer cells are a type of lymphocyte that are formed in the bone marrow. After they're formed in the bone marrow, they migrate to the tonsils, lymph nodes, and spleen, where they await activation. Upon stimulation, the natural killer cells secrete several cytokines that trigger the acquired immune responses by macrophages and other immune cells. While those events develop, the natural killer cells move into the blood and lymph, where they act as potent, non-specific killers of tumor cells and virus-infected cells. Natural killer cells are not phagocytic. Rather, they contain on their surfaces a set of special receptor sites that are capable of forming cell-to-cell interactions within a target cell. If the receptor sites match up with a group of class I MHC proteins on the target cell, the natural killer cell recognizes the target cell as one of the body's own cells and leaves it alone. In contrast, when a cell becomes infected or becomes cancerous, these MHC proteins are altered. Altered MHC proteins signals to the natural killer cells that something has gone wrong. The natural killer cells then respond by releasing cytotoxic mediators, including perforins and granzymes. Perforins effectively drill holes in the target cell, thereby destroying it. Granzymes induce the target cell to undergo apoptosis. The DNA condenses, the cell shrinks, and breaks into tiny pieces that can be mopped up by wandering phagocytes. Natural killer cells have the ability to destroy any cell that they see as abnormal. This includes both tumor cells and virus-infected cells. Complement, also called the complement system, is a group of nearly 30 inactive proteins that circulate within the bloodstream. If a microbe penetrates the body, complement proteins become sequentially activated through a cascade of steps that assist in the inflammatory response and phagocytosis. Additionally, complement proteins in the cascade bring about the destruction of the invading bacterial cells or viruses through cell lysis or virion lysis. Macrophages, dendritic cells, and epithelial cells recognize pathogens by identifying unique, highly conserved microbial molecular sequences that are not found on host cells. This system of recognition is called pathogen-associated molecular patterns, or PAMPs, and includes small molecular sequences, such structures as the lipopolysaccharide layer of gram-negative cells. PAMPs are recognized by protein receptors called toll-like receptors, or TLRs. TLRs are present on macrophages, dendritic cells, and endothelial cells. The TLR family in humans consists of at least 10 members, each working to mediate a specific response to distinct PAMPs. Specific TLRs are located on the immune defense cell's plasma membrane and the phagosome membrane. Virus-infected cells produce a set of cytokines called interferons. Interferons alert surrounding cells that a virus is present. Thank you for watching.